Okay. Last time we discussed DNA replication. So we have completed DNA replication process. This is the last lesson for the DNA replication. Now in this lesson, we're going to look how accurate is this DNA replication process. So mainly the DNA polymerase enzyme responsible to copy one DNA molecule into two. So the majority of work done by the DNA polymerase enzyme. When it is copying one DNA molecule to make two DNA molecule, at least a human DNA molecule in a cell has nearly 3.2 billion base pairs. 3.2 billion base pairs. So then 3.2 base nucleotide should be accurately copied to make a new DNA molecule. Now the question is it 100% accurate or it is less than 100% accurate? So this is a quite interesting and big topic in molecular biology, but here we're going to look a summary of this process. This one most of the time referred as fidelity. The fidelity is accuracy. What is the accuracy of DNA polymerase? Now we're going to learn the accuracy. If it is not 100% accurate, whether we are ending up many errors, or if there are errors, is there any way to correct these errors? Okay, just to give a summary of this fidelity or accuracy or errors, when DNA DNA polymerase copying DNA molecule, there is an error rate of 1 in 100,000. That means every 100,000 base copied, there should be one base error. 100,001 base error. Of course, we can calculate to the total number of bases because 100,000 is 1. Then how many? 3 billion. So we take roughly 3 billion. 100,000 for 1. Then what is the rate for complete DNA molecule? That is 3 billion bases, right? This is the error rate of DNA polymerase. But there is something very interesting thing. DNA polymerase, so far, we have learned that it just adding the bases to the complementary sequence. But it has another activity. So this is also we're going to learn here. This activity, DNA, is called proof reading. It is something like this. If we write a one-page document, let's say a letter, so we can, let's say email, just we write an email. After the last sentence, we send this email. But we have an experience, before we send this email, and this email is important email, we read it again. when we read it again, there are a lot of chances that we can find a mistake. It could be spelling mistake, it could be grammar mistake, it could be something, the meaning is wrong in the sentence, like this, we can find it. So this is the actually concept of proofreading. That means DNA, when it copies this DNA, it can bring a nucleotide and then it can add this nucleotide to the position. At the same time, DNA polymerase can check whether this is correct or not, but this can done after addition the base. When the base is added, it make a phosphodiester bond. For example, okay, we come to detail later on. So just we get the idea of proofreading. Proofreading is DNA polymerase add the base to the developing strand. After adding, immediately it check whether this is correct one or not. So then if it is incorrect one, it can repair, it can bring the correct one to the position. So proofreading is a very powerful mechanism. Why is that? Proofreading bring these error rates to a very less number. So this can bring into 1 into a billion. So here reduced error 1 into billion. That means billion bases, only one error can come. So this is still done by the DNA polymerase, right? So DNA polymerase is the enzyme done this process. Now it come to a 1 into billion. So theoretically we can say the human DNA has a 3 billion bases, then only 3 errors, 3 bases wrong. That is very less. That's very little. But still, this is not actually the exact number. That means the people who discovered this mechanism, they might say, okay, guys, this is not exactly a 1 in 100,000. It could be this much. The range, 1 in 10,000 to 100,000. And this one is actually this is 8 to 10, right? So this should be corrected as 10 billion. Just small mistake here, 10 billion. So this could be like this, a range. So I took the upper limit, right? The 10 billion is the upper limit and the 100,000 is the upper limit. That's a range. It's not exactly we don't have one number. It is a range. So this is the range we use. In this range, there could be a 1 error and here 1 error. That means at the end, when the DNA molecule is synthesized, it is not 100% same as the original one. There could be some bases. They are not same as the original molecule. That's naturally happening. This is the real situation of DNA replication. There are few, we could say at the moment error, it's not same as the original molecule. There are some bases are not correct. Still, this is the first method we going to learn, proofreading. Then there is a second method. The second method is we can always see the bases are not correct. So bases are not correct mean, let's say we can find a situation like this. G pair up with T now. Newly synthesized molecule, we can find this base pair. But this base pair is mismatched. This is not the case because G and C, the correct one is a G and C, guanine and cytosine. But guanine and thymine here, this is a mismatch. Then the second method used by the system, mismatch repair. Proofreading is the first method. It's already done by the DNA polymerase. And there are another mechanism called mismatch repair. And third one, this could be happen immediately after DNA replication, or it is existing as a continuous mechanism to keep the DNA molecule as same as original molecule. The third method is something important for long term. That means once DNA molecule is synthesized, proofreading and mismatch repair are immediate process. They try to correct it. From that point onward, proofreading cannot done. Mismatch repair is also not done. But after that, there is a continuous maintenance process. They try to correct if there is a later change, damage, or not same as the original molecule. These come into action to repair the process. So this is called nucleotide excision repair. So altogether, we have to look into three mechanisms of DNA repair: proofreading, mismatch repair, nucleotide excision repair. So let's one by one try to understand this, and we will little bit at the end discuss what happened if there is an error or there is a few errors than the original molecule. So we're going to look these two in this lesson for DNA repair and its significance. Is it clear? Do you have any questions?
No.
Okay. So it's better that for learning purpose, we have to clearly separate the three mechanisms so we can remember the three mechanisms: proofreading, mismatch repair, nucleotide excision repair. First, we're going to look the proofreading of DNA polymerase enzyme. Right. For that, I have taken some of the pictures, which is much easy that if we take these pictures, can reduce also time writing too many things. Now, what we're trying to learn, okay, let's come back to the picture later. We know that this is the template strand, and then the newly developing strand. So it is adding a nucleotide this direction. So this direction can be specify if this is a 3' to 5', new nucleotide adding to the 3' position. That means this is going this direction. So after done this much of, let's say, at least 100 nucleotide this place, 100 bases. Now here DNA polymerase going to add, this is a DNA polymerase, going to add the new base to this position. Let's say this position has G, guanine. Now the correct base is cytosine C. Now it bring here incorrect nucleotide T, and then added to the position. It's growing like this direction. Now this is an error, G pair off with T. Right here in this picture, again, you can see this is a developing strand here, 5 to 3 direct, 5 prime to 3 prime, and T is coming to the G position. Now this is already attached to the phosphodiester bond is made. So 3 prime to 5 prime phosphodiester bond is formed. Now in the next turn, it check whether this is correct sequence or not, correct order or not. That is the proofreading. So it is immediately done after phosphodiester bond form, before it brings the next one. Now in that case, when it realized this is mispaired. Now here you can see T and G is mispaired one. That is identified during the proofreading process. During the proofreading process, mispaired bases identified. Then the next is DNA polymerase is an enzyme. So this enzyme has several activity now. Now the second main activity is it can form phosphodiester bond. Now it has another activity to hydrolyze phosphodiester bond as well. It can form, it can also break it. So then it realized this is a wrong pair. Only option is immediately the phosphodiester bond should be hydrolyzed. Just should be cut from this, right? Now that's very important thing. So it used this 3 prime to 5 prime direction. This is called exonuclease activity. This hydrolyzing of this bond is called exonuclease activity. Now here you can see the DNA polymerase has to do opposite function. Immediately hydrolyze the phosphodiester bond and remove this thymine from this position. So this is a very important thing to remember. It has this 3 to 5 prime exonuclease activity. Exonuclease activity is hydrolyzing the phosphodiester bond. After that, we have to believe DNA polymerase will not make again mistake. So it will bring the correct bases to this position, and then as same as the original function of DNA polymerase. So it can bring cytosine this position, and the mispaired base are corrected. So this is the proofreading ability of DNA polymerase. This is a very important process because it can reduce in a bigger way the errors of DNA replication. So then after that, its job is done once it complete the replication process. DNA polymerase, once it completely copied the DNA molecule, its job is done. But as I told you, as we mentioned here, there is still some errors. So 1 in 10 billion or within the range of this, there could be certain error persist. Proofreading is not 100% efficient. It can still leave some kind of a mismatch. Then DNA molecule is formed, and after that it is further subjected to a check quality level, quality control level, whether this is completely correct or where there are any mismatch. So the mismatch repair process is coming for the next event. After proofreading, DNA molecule is copied and mismatch repair process is coming. So let's see second process, mismatch repair. So I should thanks for these pictures taken from this Khan Academy website, because they are good website. Can recommend for student to read. You can really use this kind of a good website. They are accurate, and the contents are correct, and they have a good explanations. Now look at here, there's a mismatch, G and T. Or maybe sometimes we can have a and C mismatch. Because there is a chance purine and pyrimidine pairing still can happen, but still, pyrimidine, purine G and A, purines, T and C are pyrimidine. So then it should be A with T, C with G. But their molecular similarity, this mismatch can happen. Now, after synthesize the DNA polymerase job is over. Then there is another group of enzymes responsible for this process, and they identify this mismatch. So it is also important, this is done with the newly synthesized DNA. It's like a quality control mechanism. When the DNA molecule is copied, the mismatch repair process immediately take into action. Let's see what is the next event if there is a mismatch. Then the second event is they're going to remove a small piece. You can see not only one. So they're going to remove certain base sequence from this place. So the group of enzymes cut this DNA molecule with its neighboring bases, right? So this is cut. So this small piece is removed, and after that, this should be repaired by, again, DNA polymerase. So DNA polymerase has a little bit of additional work after that to help to build up this replacement DNA. And then it can make it A with T. So this is mismatch repair. Okay, now the third one we discussed, this is kind of maintenance work, continuously doing the process, nucleotide excitation repair. Okay, not only changes happen during replication. After replication, changes happen in the DNA molecule. So these changes are many different changes. So we collectively call them damage. Collectively call them damage. So we can understand the original replication process. The main thing is mismatch. So incorrect base. But now we are talking of another thing, not only incorrect bases, we're going to talk about other damages. So one of the common damage, I mean, we cannot highlight everything. One of the common damage can happen, exposure of the DNA molecule to unfavorable factors can alter structure of the DNA molecule. Example, radiation, X-ray and UV type of radiation are very harmful to the DNA molecule. Or chemicals. So radiation and chemicals are considered as well-known factors which can still damage, which can still change the structure of the DNA molecule at any point. There's a one common identified factor. In this case, thymine dimer, formation of thymine dimer. So if the two thymine molecules are close together, so they allow to make some kind of a bond between these two thymine bases. So this can cause some kind of this alternation of the DNA molecule. So this is called helix distortion. Helix this is due to UV radiation. So this is not again favorable conditions. This can damage the DNA molecule as well as it's a gene expression. That means copying the genes into protein process is also altered. So then there should be again a process to repair this kind of a damage. Right, next. If this happen, so it just makes some kind of a alternation on the molecules. So it forms these bubbles as a result of this thymine dimer or helix rotation, distortion. Then what's going to happen, a group of enzymes, they're going to cut this piece as it appears on the bubble at this two end, it's going to cut this DNA. And after that, DNA polymerase going to make this new strand again. Should make it and then the ligase going to seal this two end. This is done by DNA polymerase. So this is called nucleotide excitation repair. So this is continuous process. When there is a damage identified, they can still correct this process at any stage. So that means mostly adult, the cells are exposed to the different radiations and chemicals. At that time, we have this nucleotide excitation repair mechanism to correct the process. As a result, again, we don't get altered DNA molecules during the lifetime. So replication done all the work to make it very correct. Still, there are some damage happen. This also will be corrected later to keep it correct. Okay, finally, we can end it up. Still, there are some errors in DNA molecule. There are repair process, there are fidelity, there are accuracy and proofreading, mismatch repair, nuclear excitation repair, all done it nicely to reduce the changes of the DNA sequence. But at the end, so if we come to this point, there is still change of base sequence of DNA molecule. We cannot avoid this. The nature cannot avoid this. There are some changes persist after DNA replication, and there are changes can happen during the lifetime. DNA excitation repair mechanism may be not efficient to correct these things, so then the damage can happen. So collectively, the whole concept we summarize into a mutation. But this is not the place we're going to learn mutation. Mutation is coming up in the next lessons. But still, we have to talk about mutation. The definition is that kind of a spontaneous, random change of base sequence of DNA molecules. And after that, if we want to summarize what happened to this organism, if there's a mutation, what is the consequences? If there's an error of the DNA molecule, what is the consequences? Now, this is very difficult topic to summarize. We can't summarize because mutation is a broad topic. So we cannot summarize consequences of mutation without knowing what kind of a mutation is existing. If we know type of mutation, then we can at least say, okay, this could happen. But at the moment, we still are in a little bit trouble. We have to summarize the consequence of mutation. We have to summarize effect of the mutations. So people can interpret differently. People can interpret some other ways. They can give a different example. So it can change person to person, these consequences. As a result, we have to be careful how we're going to make this mutation effect. Basically, we can say no effect. Now, if you want to understand no effect, you need further understanding of the genetic code in the next coming up lesson. Then only you understand, okay, if change happened, but there are no effect. This can be only explained after learning the genetic codes. Okay, there is an effect. This effect is Again, we divide into two. One is harmful effect. Other one is a beneficial effect. What is harmful effect? This is